Hey, welcome to our first tech talk. So first we'll be looking at an overview of how we study proteins, and then we'll specifically be looking at a biochemical method to study proteins, looking at chromatography. There'll be 4 tech talks throughout the semester, 1 for each unit. So you should listen to the audio lecture, look at the PowerPoint slides, and then there'll be a short quiz that you take through the D2L class. Okay, so if we look at how we scientists study proteins, cells, and organisms, there's 3 different approaches: biochemical approach, genetic approach, and microscopy-based approach. Okay. With a biochemical approach, what we're going to do is we're going to take cells or tissues and we're going to grind them up, and then we're going to run them out. And we might run them out in a column or onto a gel and then transfer that to a membrane, or we might use dishes coated with antibodies, right? So chromatography would be using columns. The Western blot is where we take the proteins, run them out in a gel, and then we transfer it onto a membrane, stain that, or use antibodies to detect specific proteins. And then in ELISA, you're using a dish with multiple wells where we've added antibodies that are specific to certain proteins, and that allows us to detect whether that protein is present or not. So as I mentioned, chromatography, we're going to look at more in this tech talk. I'm planning on for tech talk 2, talking about ELISAs. So we probably won't talk specifically about Western blots, but as you guys are looking for primary literatures on your pet proteins, maybe you'll find a figure that talks about Western blots. So, if we want to have a class discussion about Western blots, we can definitely do that on the fly. Okay, so looking at these other techniques with genetic approach, this is where we're going to be looking at the genes, the DNA that code for proteins. And so this is where we could take that sequence and we could mutate it, and then we could look at the phenotypes that occur. So either the phenotypes, at a cellular level, or at a tissue level, or at an organism level. And we could also, besides just making mutations where it's like a single point mutation, we could do whole knockouts, right? So we have knockout mice in which we can study the function of a protein because we've removed that gene, we've removed that protein from being able to be expressed. Or maybe we can change the expression rate because of a mutation that we've added in, or we can make the protein go to a different location because of mutation. So in the chapter 4 and later on in the semester, the idea that sequence, that primary structure, will indicate how that protein ends up folding, and how proteins fold relates to their function, right? And then lastly, we can use microscopes in order to look at proteins in cells and organisms. So we can do antibody staining to look to see where the proteins are present, or we could use fluorescent dyes. We can also use contrast in order to do that. We can do live imaging, or we can do imaging where the tissues have been fixed, or we can do scanning electron microscopy or transmission electron microscopy, right? So there's lots of different microscopy approaches. So Unit 3 will probably look at a genetic approach, maybe these knockout mice, and then Tech Talk 4, which is in Unit 4, we'll look at some of these microscope-based approaches. Okay, again, you'll see these different approaches, biochemical, genetic, and microscope-based, in any of the primary literatures you're looking at for your pet protein. And so try to be mindful when you're looking at these papers, you're looking at figures, what technique are they using? Why are they using this approach versus that approach? So keep in mind, with microscopes, those can be very expensive, so having access to a microscope might be limited, where having access to the tissue culturing where you can grow the cells to do Western blots or ELISA might be a little bit more cost effective. So a lab might opt to do a biochemical approach. Doing something like mutating of genes, the the cost-wise of doing the mutations isn't so costly. It's more having people skilled enough to do that. But then when you move into like making a knock-up mouse, that becomes much more expensive. Being able to house that organism becomes more expensive. So the biochemical approaches tend to be the the go-to approach. So that's why I'm okay giving 2 tech talks to that. My background, I did a lot of microscopy work, both in my undergrad, but also in grad school and in my postdocs. And so I love microscopy. So if you have questions about microscopy, please ask. I've used both the transmission electron microscope at TEM and SEM. I did lots of fluorescence work, and so I love the visual that microscopy gives you, right? So that piece is missing in the biochemical approach, right? You're not seeing where the protein is because of how you purify the sample. You can tell where it is, but it's not that visual of seeing a whole cell intact and seeing the location that it's at. Okay, so that would be one of the kind of downsides of the biochemical approach. Okay, so we're going to look more at chromatography. First, we'll look at how do we grind up these cells so that we can use them for biochemical approaches. So how do we get the proteins out of the cell? Well, we have to have a cell suspension or tissue to work from. So if we have a cell suspension, then maybe we've been growing these in tissue culture. So like in the lab, if we have tissue, maybe we got a biopsy from a specimen. And so we're going to need to make sure that we break these cells apart if we're talking about a tissue, or, if we've got a nice lawn of cells on the tissue culture dish, we need to make sure that they're separated into a suspension. And then we want to make sure that we're going to break the cells apart so all the material inside the cell is going to be released into this solution. Okay, so what we're going to end up creating is a homogenate, okay, and so, or an extract. And so in order to do that, to create a homogenate or an extract, there's 4 different common methods in order to do that. And you can see that in that middle of that figure. We have these 4 different methods. Okay, so one method that's used is by taking the cells and treating them with high frequency sound or an ultrasound, so that can help to break apart the cells, and so the cell membranes are destroyed, so all that material that's inside the cell is released. Okay, specialized piece of equipment, so probably not as common. The more common method in order to create a homogenate is to use mild detergents. So this is where you might add like a tween solution to the cells. That tween is like a kind of brand name of detergent. It's going to be able to punch holes in the cell. And this is why washing your hands is helpful, because when you wash your hands, you break holes into any kind of bacteria cells. You also disrupt the lipid layer, the viral envelope that's on viruses. And so what happens to that bacteria or virus in your hands is it breaks that membrane and everything inside kind of breaks out, and so they die. Okay, so wash your hands. But in the lab, when you want to create these homogenates, you add the detergent, and it's going to break those cells that you're studying apart, and the material inside is going to be released. Okay. Okay, for a third method, what you can do is you can force the cells through a small hole using pressure. And so sometimes this is done with like an apparatus that kind of looks like a syringe with what almost looks like a needle connecting it to another syringe. So you just pass it back and forth from the syringes, but that needle is very small, and so the cells are being forced through that hole, and because of that, the membranes get disrupted and the material is left behind. So then you collect that material and you can use that for your homogenate or extract. The fourth method is where you would use like a plunger, and by rotating that against a wall, because when it rotates, it hits the cells up against the wall, that causes it to lyse. Now, that plunger the fourth method there, it's kind of like a mortar and pestle. So like if you had a tissue, you could put it in a mortar and pestle. If anybody cooks, you might have a mortar and pestle where you ground like herbs up to release the oils of the herbs and to break it down. So it's the same kind of idea where you're taking this mortar and pestle and grinding it. So you're grinding these cells. So they do make disposable setups where you have this pestle and you put it into like a microcentrifuge tube and you can grind it that way to help dissolve it. Usually you would also add a detergent to that, so it's a combination of the second method and the fourth method that allows for the really breaking up of those cells so that you get the homogenate. Okay. So again, method 2 and method 4 are the most common ones that you would see like in a lab to do that. And again, usually it would be a combination of the two to really ensure that you're getting good lysing, breaking up, grinding up of the cells. So once you have your lysate, your next step would probably be to centrifuge. Okay, and so the reason why we're going to be centrifuging is we want to make sure we're isolating proteins. So, if we think about our bodies, we're made up of a lot of water, and same thing with the cells. And so we're not looking to collect all of that necessarily. A lot of times all we want to do is look at the protein fraction, right? And so if you look at the middle of this figure, we have this homogenate before centrifugation, and you can see all that liquid and all that material in there. And after centrifugation, what happens is any of the smaller, less dense components, kind of like the liquid of it, is going to stay up towards the top. And then what we would have is the pellet, and the pellet's going to be where our denser material is. Now, what ends up in the supernate versus the pellet depends on how fast, with what force was exerted on the homogenate, right? So if we spin this really fast and there's a high force, smaller components would move into the pellet, okay? So that's really important because we are actually then have an ability to differential centrifuge. So we can create different pellets. And so our next slide shows us this concept. Okay, so we're starting here with our cell homogenate, our extract. Maybe we've added detergent, we've grinded it with our disposable mortar and pestle. So if we put it under low centrifugation, what's going to be in our pellet are larger structures. So we might have nuclei, we might have cytoskeleton, but in that supernatant, some smaller organelles might be present. Okay, so our before centrifugation, this would be a whole cell lysate. With centrifugation, even at low speeds, we're starting to separate out parts of the cell. If we increase the speed, we would then get mitochondria and lysosomes and perisomes. So these are smaller organelles, but kind of in the mid scheme of the size of organelles, but definitely smaller than what we would see in the first fraction. So in the second fraction, again, we're still going to have material in our supernatant, but if we take that supernatant and increase the speed, meaning we're increasing the force, we would get further separation, and this is where some of the fragments of the endoplasmic reticulum and other small vesicles would be found in the pellet. If we take that supernatant and move it into an even higher speed centrifugation, what we would end up in our pellet are very small components like ribosomes, right? So ribosomes are protein, RNA based. There are subunits that are coming together. We also have viruses, and if we think about viruses for students who've taken microbiology, but also real-world application, if we think about viruses, they can range in sizes, right? And so they're relatively small, much smaller than most cells, and so they are going to, if we're only centrifuging at centrifuging at low speeds, they're going to be in that supername. But by the time we move up to these high speeds, we can get the viruses. And then some of these larger macromolecules would be found in this fourth pellet. So if we collect these different pellets, we then could look through biochemical analysis at where are different proteins found, right? So this technique is not, differential centrifugation are not completed all the time, but if you definitely want to show that, oh, I have a protein that is becoming activated in the mitochondria, you're going to want to make sure that you do a mid-speed centrifugation. I just briefly want to talk about the technique of centrifugation. So you can see that we have these different speeds. So low speed centrifugation would be something that you could do like on a tabletop centrifuge. So maybe one of those like little bench ones that you might have in a lab. You can put like 20 microcentrifuge tubes in, and you would spin that for like 5 minutes, right? And you would get your pellet formed. Medium speed centrifugation, this would require a larger centrifuge than that for the most part, unless you have like a high capacity one. So these ones would be like the size of like a microwave, but even in some cases they might be something that's like on the floor. So maybe, about the size of like an oven, again, depending on how old your centrifuge is. The newer ones are smaller than the old-fashioned ones. With high-speed centrifugation, these are the ones are definitely going to have warning labels on them to be extremely careful. Things at high rates of speed can harm you if not properly balanced and such. And these ones, again, are usually larger ones that would be standing on the ground, not on the bench top. Okay, so the other thing to keep in mind is that with low speed, these centrifuges are cheaper than your high speed centrifuge. So a high speed centrifuge has to be able to be balanced and be able to spin fast enough in order to get that separation. And so the technology in order to do that costs more money. Okay, so most like cell biotype labs, most labs are definitely going to have a low speed and mid speed. Usually your high speed centrifuge is shared equipment in a lab space. So some of the different characteristics of proteins allow us to be able to separate and purify them. So as you're learning about proteins in chapter 4, proteins are very diverse. So their sizes differ, so like how many amino acids are making them up. Their shapes differ, so how they end up folding together, whether they're single subunit, a single domain, or multiple domains coming together, multiple subunits coming together. How those amino acids are arranged relates to whether they're hydrophobic or not, and so whether they're positively charged or negatively charged, how those amino acids get folded in. So we might have a protein that has positive charges on the outside, or we could have a protein that has negative charges on the outside, right? And so all of these different properties can be utilized in order for us to isolate them so that we can then study them individually. Because the reality is, in a cell, we have this mosaic of different proteins that are present, and these proteins are interacting. And so if we want to study those interactions, if we want to study where the protein's located, we need to be able to eventually separate them out. So even if we pellet the different organelles, so maybe we have a pellet that has ribosomes in it, but then we have a pellet that has mitochondria, and then we have like a whole cell, we still need to be able to see those proteins, or maybe separate those proteins to see what other proteins they're interacting with. But we can take advantage of those characteristics. So with column chromatography, what's being done is that we're going to have a column that's going to have a matrix in it, and we'll talk about this matrix in a little bit, because depending upon what the matrix is, it's a different type of chromatography. Okay. So with column chromatography, you have your matrix, and you're going to apply your sample on top, and then generally it's just gravity that's going to be doing the work, so that the sample is going to move through that matrix, and at the bottom we're going to have where the material is going to leave the column, and that can be collected in a test tube. So over time, we can collect the material that's being released by the column, and we can even create different fractions or alutes to then look to see where does our protein of interest come out in. Okay, so we can see in this figure how the sample's been applied, but as it moves through the column, we can actually see there's different material in the column, right? So we can actually see there's some separation. So we've got our green protein, our red protein here, and as time goes on, maybe the red protein all runs out, so we've collected that, but it takes longer for the green one to run out. Or depending upon what's in this matrix, we may need to add a different solution to release the green protein or release the red protein, right? So these might all become bound, and when we add a certain solution, the red one comes off. When we add a different solution, the green one comes off. Okay, so depending upon what, again, the matrix is, kind of changes how the technique is done, what material is going to come off when. Okay, but again, the idea is that we have this, again, mosaic of proteins that are present that we're putting over on this column through the sample, but we're able to actually isolate the different ones. We then could use these different fractions to do additional tests, like a Western blot, like an ELISA. So as I mentioned, the different material that makes up the matrix will give us different types of chromatography. So this figure looks at 3 different types of common chromatography that are utilized. So we can have ion exchange chromatography. This is where we would have beads. These beads are positively charged. So the positively charged beads would then attract any negatively charged proteins, molecules that are present in that sample that you've added. So as the material flows through the matrix, your positively charged molecules and proteins would be collected first. You then could add a solution that would maybe change the pH, and that would allow for these negatively charged molecules to be released by those beads, and then they would move into it. So you would switch to your test tubes before you added your solution. Okay. Another type of chromatography is gel filtration chromatography. And so in this case, we're going to have beads that have holes in them, and those holes can be different sizes. And so the proteins, the molecules, have to be a certain size in order to get trapped into the beads. So that means larger molecules would be able to move through that gel. They would not be trapped in the beads, so they would be collected first. You then would add a wash, you would add a solution that would allow for the release of those molecules in the beads. Okay. The last chromatography we're going to talk about is affinity chromatography. So affinity chromatography uses beads that traditionally would have antibodies attached to it. Sometimes there could be enzymes that are attached to it instead. We're going to focus more on the antibodies just because that's my background and my area of interest. And in the chapter 4 lecture slides, I have more slides about antibodies than other proteins, so to kind of make that link. Okay, so again, these beads are going to be coated with like an antibody, and again, in some cases, maybe an enzyme. So it would recognize those specific molecules that the antibodies meant to recognize, or the substrates, which the enzymes would recognize. So other proteins, other materials would be able to pass through the matrix, but your protein of interest that the antibody binds would be trapped, right? So again, we could then add a solution that maybe changes the pH, so it would denature the antibody, not allow it to bind and release, or sometimes a high salt concentration is added. Different material, different solutes can be added that would release that bound. Okay, so I'm just going to have a couple more slides just to talk about affinity chromatography. So again, affinity chromatography, we're talking about an antibody being bound to the bead. We'd have a bead at the bottom of this antibody, and so keeping in mind the antibodies are going to bind specific antigen, that they have antigen binding sites. So if that antigen, if that protein is present in our cell lysate, our homogenate, or extract, it would get trapped and bound until we break that bond. Okay. So this figure is just showing you the idea of, again, of affinity chromatography. So we would have antibodies that would be specific to a specific antigen, right? And so these are anti-A antibodies, right? In this mixture here, the molecules, that would be like a homogenate from our cells. So we've ground up those cells. Maybe we added detergents. Maybe we used the mortar and pestle to create the homogenate. So when we pass a homogenate through this column, it has these beads that the antibodies have been bound to, right? So the whole column is going to be packed with these beads. When we add the homogenate, all the other molecules, all the other proteins are going to be able to pass through, but the antibodies are going to catch all of them. So in this example, the antibodies are going to catch all the A's, so all the other letters are allowed to pass through. Once we add a solution that allows the antibody to stop binding, and usually that's done again by changing the pH or salt concentration, because that denatures, it changes the shape of the antibody enough that that binding site's no longer available. We now can collect that pure antigen, and again, for this example, our A's, right? So all the A's are going to be collected here, and we then could follow this up with by doing a Western blot to make sure it's pure, or some other technique. Or maybe we're going to be using this sample with purified antigen as a vaccine, so we're then going to immunize individuals with it. So if we kind of take this to a real world application, what we could do is we could take, COVID, and we could infect respiratory cells with the COVID. That would grow up lots of the virus, right? So we could take the virus, we could homogenize it. So we would have viral lysates, if you will. If we take samples from people who've recovered to the convalescent plasma that's going to have antibodies, we could create a column with that plasma. We could pass that viral lysate over, right? And so all of those proteins that, at least that individual with that we got the convalescent plasma from, all those proteins that their immune system did not respond to, would flow right through, right? But the proteins that are specific to the antibodies would get trapped. So we could release those, and then we would know which proteins, at least for that individual, were good targets from the immune system, right? Because these people recover, right? Their immune system responded, made antibodies, they got better, right? So we have to make the assumption that their immune system picked good targets. So that's a starting spot, right? That's somewhere to move next step, right? So we could take those antigens and then we could say, well, does that make a good vaccine candidate? Could we immunize individuals with some of those viral proteins, right? It's not the whole virus, it's just proteins that the virus would make. You got infected, and allow your immune system to make antibodies against it, so that if you were exposed to COVID, you would then have protection against it, right? So there's definitely, researchers working in labs like Emory and, Harvard and all over the country, all over the world, that are looking and using this technique in order to try to determine what are good targets for vaccines. Okay. So that concludes our first tech talk. If you have any questions about the material that's in the tech talk or the PowerPoints, please send me an email, reach out, ask questions on Monday during our class. I will be posting a quiz with material that's contained in the PowerPoint and also in this audio lecture. So thank you for your time.